Okay, let's finish up chapter 6, talking about protein stability and trying to figure out what makes proteins stable. And hopefully, we'll make the case that it's the hydrophobic effects. And secondarily, there'll be some electrostatic interactions. Also, talk about denaturing proteins and how you can renature them or refold them. And what we need to walk away with is the protein structures are flexible and may include unfolded regions. So there's favorable interactions in proteins. You have the hydrophobic effect. And the key of this is it's the release of water molecules from the structured solvation layer around the molecule as the protein folds. And this increases net entropy. So, in other words, what happens when you put a nonpolar molecule into water is the water tries to solvate it the best it can. There's not favorable interactions between the nonpolar group and the water, so the water ends up forming a cage-like structure or an ice-like structure around that hydrophobic group. And you pay an entropic penalty for that. That's a very organized structure. You're not getting favorable interactions between the water and the hydrophobic group, and then you pay this entropic penalty for ordering the water. So as you remove those nonpolar groups from the water and they associate with each other, you have less water nonpolar interactions, and then the water is free to be disorganized, and that is more entropically favored. You do get hydrogen bonds between the hydrogens off of the amide nitrogen and the carbonyl. These are a lot like what you see in DNA. Excuse me, they lead to specificity, but not a whole lot of stability. And the logic behind that is that you can hydrogen bond with water, or you can hydrogen bond within a protein, and energetically you'll have the same amount of energy. You're not really going to pick up more hydrogen bonds within a protein. But those hydrogen bonds can stabilize structures like we saw in the alpha helice. Okay, you also have London dispersion forces, which are medium range, weak attraction between all atoms, and they contribute significantly to the stability of the interior of a protein. So you get these nonpolar residues to come in to close proximity with other molecules, and those can actually be quite significant, but really the driving force for that is trying to get those hydrophobic groups away from water. And then finally, you get electrostatic interactions. And these are long-range, strong interactions between permanently charged groups, sometimes referred to as salt bridges. And these salt bridges can sometimes be buried within the hydrophobic environment, and then they'll stabilize the protein. The issue with these electrostatic interactions is they could also be involved in hydrogen bonding with water. And then you question, well, do I get a lot of stability if I'm hydrogen bonding with water versus forming a electrostatic interaction between charged groups? So here's just a little diagram showing all the type of interactions that you can get. But we find out the proteins are only marginally more stable than their unfolded form. And the reason that they're marginally stable and not rock solid stable is the proteins still need to move. And so too much stability, meaning everyone locked in very fixed positions, would work against catalysis or other functions of the proteins. So the proteins need to be able to flex and move. And so the free energy required to denature proteins around 0.4 kilojoules per mole per amino acid residue, meaning that per amino acid residue, you're not getting a whole lot of stability. Remember, by comparison, it takes 20 kilojoules per mole to break a hydrogen bond. So overall, the overall energy you get per amino acid residue is a fraction of what you would get if they were just all hydrogen bonding in the absence of water. So interactions that stabilize proteins, you get disulfide bonds, and you can see an example of those right here. You get hydrophobic or van der Waals interaction, London dispersion forces that you can see here. You get hydrogen bonds, and you can get those hydrogen bonds between strands, like in a beta pleated sheet, or alpha helices. You can see those. Or you can get them between different backbones, such as in terns, or you can get them between different side chain residues, or between a side chain residue and the backbone. So all different sorts of ways to get hydrogen bonds there. And then you get these electrostatic interactions between the positively charged amino acids like lysine, and then the negatively charged amino acids like aspartic acid. So those are the forces that that are involved in stabilizing a protein. But we still want to answer this question: why are proteins only marginally stable when evolution should seek to produce a super stable structure? Again, well, proteins need to move. And again, this is repeating the previous one, but it's important. Too much stability would work against the catalysis. You'd lock this into a rock solid foundation. And again, we're getting marginal stability per amino acid residue. So each of these non-covalent forces has energies that could total thousands of kilojoules per mole over an entire protein structure, if you looked at all the possible hydrogen bonds. So that means that we must be balancing some unfavorable interactions with favorable interactions, but not maximizing those favorable interactions. Again, if you maximize those favorable interactions, you're going to make a protein that is just rock solid and doesn't have much conformational mobility. And the other thing that provides that marginal stability is many of these non-covalent interactions, especially the hydrogen bonding, but also the electrostatic interactions, can form with water if the protein's unfolded. So we need this delicate balance among these powerful forces. But what we find really is it's a hydrophobic effect. So probably the greatest influence on protein stability. And again, stressing that the water minimizes the contact surface area with the nonpolar solutes. And so what you can do is actually measure the hydropathy of an amino acid, essentially its tendency to associate with nonpolar groups or with water. And so you can see that isoleucine and valine and leucine and phenylalanine and methionine and alanine, those are all positive values. And then we look, and as we increase the charge and we get to arginine that has a guanidinium group out there, it tends to have the least. And so this just means that these would have the most favorable interactions with water, and these would have the least favorable interactions with water. And so what you can do is measure this hydropathic index. So what they do is they kind of do a running total over 9 residues here, and then they sum it up, and then they move over residue and sum up the 9 previous. And so what you can see is that regions oscillate in the proteins. Now, these bands up here indicate the interior of the protein, and these lines down here indicate the exterior of the protein. And that comes from 3-dimensional looking at the structure. And so really the take-home message here is you look and you see that most often we see these hydrophobic peaks appear on the interior. Now that's not always true. Like this one here, some of it appears on the exterior, and you notice here is a big interior portion, and the amino acids aren't all hydrophobic. But you can see down here that we see very hydrophilic residues on the exterior. Again, this isn't perfect, but you do see this oscillation, and it does match pretty well with where those residues are found, either on the interior or on the exterior. There's other things that you need to consider when you pack these residues together in folding a protein, and that is, well, how sterically compatible are they? Am I trying to put 2 large isoleucines in the same area? Well, that's going to be bad. So maybe an isoleucine and an alanine would be better, having a large amino acid and a small one there. Where if I had an alanine and a glycine, that could be bad, because then I actually have a gap between those side chains, and that could allow water in. So we need to balance those steric compatibilities with how much volume they actually have in there. So the hydrophobic seems to be the major, but ion pairing or salt bridges can also be very important. So here's some data. So about 75% of the charged residues in a protein are involved in an ion pair. So that lets you know that just having a free ion, either associated with water or just by itself, isn't as favorable as having it paired with another oppositely charged side chain. And even these ion pairs are actually found on the protein surface, where you think they could actually exchange or be involved in hydrogen bonding with water. Ion pairs are poorly conserved among homologous proteins, and so what that means is that you can get rid of them and the protein is still functional. So that means that they're not absolutely important for the function of proteins. So they could provide some stabilizing force, but they're not necessarily conserved, meaning that they're essential for that protein's function. So these charge-charge interactions are favorable, but you do lose some entropy when you start locking these groups into salt bridges. They don't have as much flexibility, so you do pay an entropic penalty for getting them involved in salt bridges. Okay, so hydrogen bonds and protein stability. So one view is the H bonds don't count much for stability, and we've said this before because those hydrogen bonds could also form with water. And so really exchanging the hydrogen bond with water for a hydrogen bond within the protein, you're not going to gain much in additional energy there. So just like in DNA, the H bonding adds specificity to the protein structure, but not necessarily stability. So if I can match up my hydrogen bonds, I can actually force my protein into a given conformation, but it's not necessarily adding stability. So there's been some studies that suggest that total hydrogen bonds contribute between -2 and 8 kilojoules per mole to the energy difference between the unfolded and folded proteins. So if one hydrogen bond actually can result in 20 kilojoules per mole, and the overall hydrogen bond contribution is only 8 kilojoules per mole, you can tell that there's not a whole lot of stability coming from that hydrogen bond. So it's really going to be the van der Waals forces, weak and effective over short distances. So the key is the protein interiors have lots of volume and they're tightly packed. And so when we pack together these nonpolar groups, it tends to be significant over the entire protein. And just a little reminder of the charges that we're talking about. Again, this looks like it'd be significant, but if you're going to hydrogen bond with water, you're going to hydrogen bond in protein, you're not really going to gain that amount. But if I can take these hydrogen bonds, or sorry, not hydrogen bonds, but nonpolar groups and get them to associate, I do get a lot, well, a small amount of energy, but if I get a lot of those, that can sum to a large amount. But the other advantage is, as I move these hydrophobic groups out of water, I really get an entropic payoff there. So there's other ways to stabilize proteins, and that's often to include metals in there. And so a lot of the side chains will chelate or act as ligands for the metal. So metals are often charged and will like donation of electrons into them. So some really good donating groups are the deprotonated carboxylic acids, aspartate and glutamic acid, as well as histidine. Histidine's really awesome at coordinating ions, and we'll see that's very important in hemoglobin. Cysteine, the thiols, also are very good at interacting with metals. And so you'll find all sorts of metals inside the interior of proteins, and that can act as a lock. And so you can see here, this is a zinc finger motif. These proteins interact with DNA, and they tend to be short peptides. So by themselves, typically wouldn't fold up into a defined tertiary structure. But once you put this strong interaction between these side chains and that metal, you do get a defined structure. And then you can actually get more complex. This is what's called an iron sulfur cluster. 4 irons, 4 sulfurs in a cluster, and then you get additional thiols from the cysteine residue interacting with the irons there. And these do some very interesting chemistry. If you want to know more, then you can talk to Dr. Pierce. This is one of her favorite things to talk about. Okay, now disulfides you think would be really good at stabilizing proteins because it's a covalent interaction. But what you can find out is that disulfides aren't always important for the folding of proteins. So in fact, what you can do is a site-directed mutagenesis and actually swap out those cysteines for a different residue, and you find out that the protein still folds up and is functional. Now, these disulfides, actually within the cells, you don't find disulfides as frequently as you do with proteins that are released from a cell. Within a cell, it tends to be a reducing environment. Outside the cell, it's very oxidizing. And also outside the cell, you get random fluctuations of temperatures and a lot more variability. So disulfides are uncommon in the cytoplasm, which is often too reducing. And we talked a little bit about that. Glutathione is found in an intracellular concentration at nearly 5 millimoles, which is a really high concentration, and that's a thiol. And what it does is it goes around and reacts with reactive oxygen species. So within the cell, you try to keep it in a reducing environment, which means that it's not very favorable for disulfides. You're going to reduce those down to free thiols. But these disulfides are relatively common in extracellular proteins, where that covalent stabilization is more important, where you may see larger temperature fluctuations, and you actually have oxidizing environments, or the pH may change. So this is just a picture of RNase A. It's an endonuclease. And so it's going to cut specifically RNA and not DNA. And you can see that it has these 4 disulfide bonds there. And we're going to talk about those in a second, after we talk about denaturing proteins. So you can denature a protein by changing the conditions that it's in. You can change the temperature. You can mechanically disturb them, but the definition of denaturation is the loss of the tertiary structure and the secondary structure. And so when you do this, the solubility drastically decreases. So when you heat up an egg, what you're really doing is denaturing the proteins. The egg white is clear, but as you heat it up, the proteins denature and aggregate and precipitate out and turn white. And enzymes tend to lose all catalytic activity when they're denatured. You don't have that organization, so they can't perform their function. So there's lots of ways to denature. And when you do that, you put them in what's referred to as a random coil, totally random conformation of a denatured protein. And so you can do this by changing the pH. And so you may question, why does the pH denature? Well, if I increase the pH, I would start to remove protons. And I may remove a proton from an amine group that is involved in a interaction with a carboxylate group, a salt bridge. And as I remove that proton, then I no longer have that favorable cation anion interaction. And so that could lead to instability. Cysteines and tyrosines actually can be deprotonated at high pH. So imagine that I had a tyrosine that was actually involved in a hydrogen bond with a carboxylic acid. So I could actually get kind of a hydrogen bond there. But let's say that I increase the pH, and now I'm going to deprotonate that residue. And that happens at a pretty high pH, around 10. But now you look, and I have a negative involved or associated with a negative, and those would repel each other. And so that's just one example of how changing the pH could cause a protein to denature. There's other things that you can add, such as urea and guanidine. These are called chaotropic agents, and we'll talk about them in just a second. But they tend to actually help stabilize nonpolar groups in water. Function's not completely understood, but by stabilizing those nonpolar groups, you can actually unfold the protein and expose those nonpolar groups to water as long as you have these chaotropic agents around there, urea and guanidine. And we'll show you those in a second. Detergents, so those are things that have polar head groups and nonpolar tails, and you can imagine that those polar head groups could associate with polar regions of the protein and the nonpolar tails with the nonpolar regions, and essentially unfold the protein and keep everyone happy because they have compatible intermolecular forces. Organic solvents, again, what those will do is allow the exposure of the nonpolar groups out into the solvent. You can heat or you can just agitate proteins. So one way to, instead of scramble your eggs, is if you just agitate it. If you make fun of it, call it names, poke it, it will get agitated, and that can denature. And I hope someone's listening and laughing, and that's not really the agitation that I'm talking about with egg whites. I'm really talking about whisking them. If you mechanically agitate them with a whisk, you can take egg whites and turn them into meringues, which is a denaturation of the proteins. Now, you got to be careful and not include any of the egg yolk, which is high in fat, because if you include the egg yolk when you're trying to make a meringue, it actually will cause the meringue to, instead of expand, your meringue will be flat. So you just want the egg whites, and you just want to denature those proteins and include lots and lots of air in there. So that's another way that you can denature proteins. We talked about it earlier, but exposing proteins to the air-water interface will cause them to denature. So salts, heat, pH extremes, detergents, pressure, or other things will all denature. And again, as the protein denatures, the hydrophobic parts come out and they start to be exposed. They don't want to be exposed to water, so they start to interact with other hydrophobic parts of other proteins and aggregate together. And when you get these stable aggregates, they can be very difficult to resolubilize. So it's really hard to take a denatured egg and renature those proteins, turn them back into an egg white. I will tell you that within the last couple of years, someone actually did that experiment. They took an egg white and a denatured egg like this, and they actually turned it back into the egg white. They renatured all the proteins. I can't remember that. Should that be the bonus? Send me the article where they did that, just the title of the article where they essentially renatured the protein. So a cooked egg, and they renatured the proteins in it. Okay, that'll be the bonus for this one. So I talked earlier about salts that you can use, and so these two common ones, again, they're called chaotropic, causing chaos is kind of their name, but they increase the solubility of nonpolar substances in water. So again, their function is not fully understood, and you actually need them in fairly high concentrations, 5 to 10 molar. That's pretty high concentrations, but they do denature, and they also solubilize proteins. So they help those nonpolar groups be exposed to the solvent, but they prevent those nonpolar groups from interacting and forming aggregates that precipitate out. So when we talk about folding, we talked about a little bit earlier about why a molecule would. Its primary structure and those phi and psi angles, the molecule's trying to find the most favorable conformation. Maybe not the lowest possible energy conformation, but a favorable energy conformation. And so there was an interesting experiment that was done. Actually, I need to go to the next one to figure out the date. So 1957, Christian Anfinsen, he got a Nobel Prize for his folding work. He took RNA ACE and denatured and then refolded it. So let's look at how he did that. Now, we looked at this molecule a little bit earlier, and I told you, actually, I think I said it had 6 disulfide bonds. That was a lie. It's got 4 disulfide bonds. It's got 8 cysteines in total. So if I said 6, I lied. It's got 4 disulfide bonds. And so to completely denature this protein, you need to add in a salt or some other way, as well as a way to break those disulfide bonds. And that's what the mercaptoethanol does. And so then you get a completely random coil protein. Then what he found is if he just kind of left it there in the presence of oxygen and removed the salt, so a little bit of oxygen, and just pulled the salt out slowly, he eventually got 100% active protein. He recovered all the activity that he had before. Now he did another experiment, and that's down this side here. What he did is he removed the mercaptoethanol so everyone could form a disulfide. Now, since we've got 8 thiols in there, you've actually got many different ways that you could form those disulfides. In fact, the chance of forming a disulfide here, yeah, don't question my statistics. I never had the class, but this is how you'd calculate it. So I got 1 out of a 7th chance that I'll form the right disulfide. Then to form the next one, I'd have 1 out of a 5th chance to form the correct disulfide. Then to form the next correct disulfide, I'd have a 1 out of a 3 chance. And then if all I have is 2 thiols left, the chance to form the correct disulfide is 1 out of 1 chance. So if I times that all together, the chance that all the disulfides form correctly is 1 out of 105. So about 1% of the protein would actually fold properly. And he actually found that when he removed the mercaptoethanol and just allowed the disulfides to form, he did get a little bit of functional protein, but only about 1%. And that is because the disulfides just formed randomly. So then what he did is he removed the urea, and again, only about 1% of the protein was actually folded up correctly. And then what he did is just threw in a minute amount of mercaptoethanol, and he allowed those disulfide bonds to be cleaved open and then reformed. So not enough to cleave them completely, just enough to cleave some of them, and allowed essentially all those disulfides to scramble till they found their ideal partner, and then he again recovered 100%. And so from this experiment, not only did he get a Nobel Prize, he came up with a dogma, which is just a way of thinking that the structure of a protein depends only on its sequence. So he says, really, everything you need to know to get to the protein's fold is just in its amino acid sequence. And so a unique amino acid sequence will fold into a unique minimum energy structure. And it does this to satisfy constraints within its own structure, meaning it needs to deal with its phi and psi angles, and it only has limited access to those angles. It wants to bury the hydrophobic side chains and form more favorable van der Waals and hydrogen bonding interactions within the proteins. So the sequence will help it fold up into the structure. But remember, proteins are dynamic. And so there's some videos down here that actually show some computer calculations, molecular dynamics of proteins moving. And so here is myoglobin, a protein, and it's got molecular motions calculated at the interval of 5 picoseconds. So you can see that these are vibrating, and you can see that some parts vibrate more than another. So these are not static, and what we mean by static is just consistent structures. They are flexing and moving, and that is thought to be very important for catalysis. So you may have heard of the lock and key model, and that kind of is not considered the way that enzymes work. It's thought that they breathe and allow their substrate in, and then these molecular motions, these small molecular motions, allow the reactions to occur. So you don't want an enzyme just to be static, because then the substrate would come in and bind and not do anything. You need groups to move and things to attack. And so it's thought that these small molecular motions allow a lot of the catalysis and the functional properties of proteins to occur. But there are some proteins that are intrinsically disorganized. And so here is an example down here at the bottom. So this is known as KID, which is a kinase inducible domain. And it's unstructured until it finds the KID binding domain of another protein. And so in solution, it's very disorganized, doesn't have a lot of defined structure. But once it hits its target, it can adopt this alpha helice here. And so it's thought that this actually disorder is beneficial. It allows it to sample a lot of conformations until it finds its binding partner. And when it finds its binding partner, it will adopt one of those conformations. And it's thought that it can be what's referred to as somewhat promiscuous. It can bind to other proteins because it's not locked in one conformation. Once it's released from this protein here, it will end up going back to this structure, and then it could interact with a different protein and maybe adopt a slightly different conformation due to it being unstructured here and only structured when it binds to a protein. So these intrinsically disordered regions tend to have those amino acids that you see at the top and tend to lack the really hydrophobic amino acids. So that's kind of interesting. Kind of suggests again that having these hydrophobic residues leads to order because you want to bury those hydrophobic residues on the interior. And in doing that, that helps define the protein structure. So that gives us our protein stability. And in the end, we just want to talk about protein folding. I mentioned a little bit about the protein folding, but we want to focus a little bit more on it. And why does a protein fold in a given way? So again, Anna Finson's dogma is all the information needed to fold a protein is in its sequence. And so biochemists think this is generally true. But sometimes proteins need a little bit of help to find their true structure. So they may get close, but they may need some help to find their true structure. But the structure does represent an energy minimum, not the most energetically favorable structure, but an energy minimum. And I'll show that to you in just a second in what's referred to as a protein folding funnel. So here's an example of a protein coming off a ribosome. And again, we haven't really talked about this process, but in the case of prokaryotes, there are proteins, what's known as a trigger factor, that's located with the ribosome. And so as the protein comes out, it helps the protein fold. And these are called chaperones. So we're going to talk about how chaperones can help fold. But first of all, let's just talk about protein folding in numbers. So does a protein just sample all possible conformations until, like Goldilocks, it finds the just right? And so there's been a simple calculation, and that is if I have a protein of N residues and it has two N torsional angles, the phi and psi angles, and let's just say those phi and psi angles each have three stable conformations. Well, sorry, they each have three stable conformations. Then a protein of N residues would have 32 N possible conformations. And if we could sample one conformation every 0.1 picosecond, which is kind of a good assumption. Again, this is just what they refer to as a back of an envelope calculation to just kind of get you the general numbers. And in fact, that's what this means here is they just down it and just say, 32, that's close to 10 N possibles. That's just to keep the calculations simple. And so again, about 1 picosecond, that's about how long it takes for a bond to reorient. It would take a protein T seconds to explore all possible available conformations. And so you can see this little calculation. So I got 10 to the N conformations. So N is the number of residues. And you got 10 to the 13 possible conformations there. And so for a 100 residue protein, the time it would take is 1,087 seconds, which is too long for a protein to fold. So I'm doing the calculation right now. So that is over 18 minutes. Our bodies need to respond a lot faster than 18 minutes to fold a protein. So it's thought that folding is not completely random. In fact, it's highly cooperative, which means that things tend to interact with other things and they fold together. But there is some hierarchy in that. It's thought that first, as the protein comes off, you get these small secondary structure. And so you can see here in this diagram that we get our alpha helices and some beta sheets. And so you get these local bits of secondary structure, and those form within 5 milliseconds. I say milliseconds. So in fact, the alpha helices can actually form as the protein's coming out of the ribosome. So those can happen really quick. And when you do that, you start to compact the protein. So these secondary structures are pretty compact. And so what they start to do is interact with other secondary structures, and the protein kind of collapses into an overall structure. Okay, and then it says the tertiary structure probably isn't initially correct. Well, if the tertiary structure isn't initially correct, well, how do you actually get a... Okay, so it'll collapse in, and likely that tertiary structure isn't originally correct. So over the next 5 to 1,000 milliseconds, the secondary structures become stabilized, and the tertiary structure begins to form into subdomains. And then over a couple more seconds, the protein rearranges its tertiary structure and finds the internal side chain packing and H bonding and kicks out extra waters to get to its overall idealized fold. So the diagram of the book implies that alpha helices and beta sheets form on the same time scale. So it often is shown that getting beta sheets actually takes much longer. So alpha helices are really quick to form, and beta sheets a little bit more. There's a little bit more organization. And you can imagine, especially if they're parallel, you can see down here that you actually need an extra long loop to make sure that you get that parallel beta sheet. And so getting that organization, you can imagine, takes a lot longer versus just kind of twisting up the backbone. But this is what I want to get to. This is what's called a protein folding funnel. And what you've got across the top is entropy, so disorder. So all possible disorders across there. And then you've got energy here. So the energy is decreasing, but as you decrease the energy, the entropy is decreasing as well. You're getting more organized. And you notice down here, you could have a lot of little places to get hung up. So if you're following this, you could actually get hung up here. And this is what's called a local energy minima. Now, this is pretty high in energy, and there's probably enough energy around that you could get over that small hump, and you may find yourself trapped here, or you may find yourself trapped here. But if there's enough energy, and there typically is in the human body, you're at 37 degrees Celsius, there's quite a bit of energy. So you can find yourself stuck in one of these, but often there are, there's enough energy that you can reorient and find yourself down here. But you can find yourself trapped. And then you can see that down here, these barriers are a lot more. This might be the native structure right here, but it could be that you get stuck over here. And that's quite an energy barrier to get back over to the most stable structure in this point. Sorry, I was using my mouse, so I don't know if you could see that, but what I meant is I'm getting stuck in these places here. And then enough energy that I can get over. But I could get stuck down in this one right here, and that's kind of a high energy barrier to get over. So sometimes you get stuck in these, and they're only slightly different conformations, and maybe it's still functional, just not as functional. Or sometimes it could be that this is a completely different function, or maybe even non-functional, and it's just slightly different in energy. but it's too high of a barrier to actually get over there. So we'll talk about that. There's some diseases that arise when you misfold your proteins. So sometimes you get these different conformations that are close in energy, and they have slightly different functions or properties or activities. So here's actually a protein folding funnel. And so again, you got entropy. So you've got every possible conformation across the top here, lots of disorder. And then as I go down, I'm getting more and more disordered. And so you can see that some of those conformations are very unfavorable and some are favorable, but sometimes you get stuck in these local minima. And either you need just thermal energy to get out or some other tricks to get out. And so some proteins don't fold correctly in cells. So you get disulfides that aren't forming in their proper orientation, or the hydrophobic parts, or getting the hydrophobic parts in the interior. Sometimes if they're exposed to the exterior, they'll again aggregate. So we showed you this before, this picture. This is what's referred to as an inclusion body. So sometimes when you express too much protein in there, the cells don't have enough time to fold it properly. And so they just kind of package it away. And the hydrophobic parts all interact. And so you get this aggregate there. So typically unfolded proteins are bad for cells. They're going to have their exposed hydrophobic parts. They're going to stick to stuff. They're going to aggregate. They're non-functional. Cells are actually pretty crowded, so they're going to be taking up space and being, again, just kind of non-functional there. And so there's a couple different things that help proteins fold correctly. Like I said, one of the problems is if the disulfides don't form correctly, you need to fix that. So on Finson, what he found when he was doing that RNase experiment, he knew that proteins, in his example, they would fold correctly, even though they had 1 in 105 chance of folding correctly. So he went looking for something that helped shuffle those disulfides, and he found an enzyme called the protein disulfide isomerase, known as PDI. And essentially, it's just like a free thiol, but what it does is it'll come in and attack a disulfide and make a new disulfide here. And then this free disulfide can come over and attack that one. And so now I've got a new disulfide within the backbone. So 2 and 3 are now connected. And then that would expose 4, and 4 could then come and attack 1 and kick out the PDI. And then if that arrangement was the more favorable one, then you would settle into that arrangement. And the PDI actually helps also form the disulfides at the start. So when they come out there, they use free thiols, but I need to actually get a disulfide. So the oxidized PDI has a disulfide. This attacks. Now you have a disulfide between the PDI and the protein. And then again, this thiol could come over here. So it's kind of random how this is working, but yet the sequence of the protein is helping it find these idealized disulfides. And if it's true, what Finson said is that the sequence of the amino acids will actually get the protein into the right conformation. And when you do that, the correct disulfides are going to be formed. Okay, so there's a box there that actually talks about predicting protein structure and actually designing them. This lecture is going a little bit long, so I'm going to let you read that. But if we have a sequence of amino acids, can we predict the structure of those amino acids? So, like I said, you can write my name in amino acids, McKay Stephenson, and I don't think that protein exists. So if I took that protein, MACKAYSTEFFENSEN, could I predict the structure that it has? And the answer is yes and no. If there is a protein that is closely related to it, I have a really good chance of getting its structure. But there's other ways if that is a completely unknown protein, and those are known as threading. I'll let you read about that, and then ab initio, which is just calculating from first principles, and that's very difficult to do because you essentially have to test all possible conformations. But they are getting better at it. And then on the flip side is protein design. Can I come up with a sequence of amino acids that will fold into a a defined shape? So can I make this structure down here? And they actually have a competition every couple years, and they give people a fold to design, and you come up with a sequence of amino acids and see if your amino acids will fold there. And they also have structures of proteins that are new. They get the X-ray crystal structure, and then they send that sequence out to a bunch of these computational chemists and say, hey, what do you think this folds like? And so it's a competition. So everyone will turn in what they think the fold is like, and the one closest to it will win. And I think they just win glory and fame. So these are kind of different sides of the same coin, being able to design proteins that have a defined structure. That's pretty hard, but they are making progress. Look at Dr. Baker at the University of Washington, I believe is where he's at. His name's mentioned in that box, doing some incredible work there. Really interesting. If I was young again, that's what I'd go into. It's pretty cool stuff. Okay, so make sure you read box 6.4, some really interesting things. We need to end this lecture here, and so we're just going to talk about two things. How do proteins that have a hard time forming their right structure find the right structure? And I mentioned that earlier, and that's by chaperones. And then what if proteins never find their correct structure? Okay, so there are molecules known as chaperones. And what they do is they chaperone proteins and help them adopt the correct structure. So there's different classes of these. This is an example of the GRO-L, GRO-ES chaperonin structure. Let's just talk about generally how these work. So chaperones prevent misfolding and aggregation of unfolded peptides. So if I haven't said it enough, I'll say it again. The aggregation tends to occur when you get the nonpolar groups exposed to water, and then they find other nonpolar groups on unfolded proteins, and they tend to aggregate. So if you think about chaperones, one of their big jobs is to just make sure that those hydrophobic groups get tucked in the interior. So they don't actively promote the folding of the substrate protein, but instead prevent the aggregation of unfolded proteins. So for any polypeptide release, some fraction are going to be in their native conformation, and others are going to be in a misfolded or slightly misfolded. And so cells contain a variety of molecular chaperones. And so let's just talk, anyway, you can look at this little diagram. We'll see a diagram later, but there's a variety of chaperones. They're what are known as the HSP, heat shock proteins. And I believe the 70 is by how large they are. And the heat shock 70 proteins found in prokaryotes and eukaryotes. The trigger factor, I talked briefly about that one. It's attached to the ribosome of prokaryotes. You get the heat shock protein 90 found in eukaryotes, and it's one of the most abundant proteins in eukaryotes, accounting for up to 6% of cellular proteins under stressful conditions. Now, the reason why they call these heat shock proteins is what they did is they took cells and they heated them up. They made them very uncomfortable and agitated them and looked for new proteins that were expressed. And so these are new proteins that were found, these heat shock proteins. And they didn't know the function at first, and then they started looking and realized that they're actually chaperones. They help proteins to fold into their native conformation, which makes sense. If you are stressing out a cell, then the cell is going to produce proteins that help stabilize its important proteins. And then you got these chaperonins, prokaryotes, and you found in prokaryotes and eukaryotes. The example that we're going to spend some time looking at is this GRO-L, GRO-ES chaperonin structure, and it's found in the prokaryotic cells. It's got 14 subunits arranged in 2 stacked rings of 7. So let's go and look at a diagram of how this operates. Okay, so we can see in this little diagram here that we'll start up here in this corner. So you've got these two rings. They're actually identical. They'll have different conformations, but what happens is you get an unfolded protein that finds its way in the interior. And there's 7 subunits around the top ring and 7 around the bottom. And in this case here, you'll bind 7 ATPs. And then you get this GROW ES cap that fits on top. And when it fits on top, it causes a conformational change and actually opens up this interior right here. Now, this is a little bit misleading. This can't transition in between those two rings there. And so as you get that conformational change, like it says, within about 10 seconds, the ring actually hydrolyzes the 7 bound ATPs and releases the phosphate there. And in doing that, when I get ATP here, that weakens this interaction and the cap will end up falling off. And then this protein will migrate. It weakens this interaction there. And then what happens is in the bottom ring, you can get another improperly folded protein and ATP to bind. And when you get those to chain or start to bind, then the GROW-S actually dissociates off the top of the top ring. And that allows a better folded protein to come out. Now, that doesn't mean it's maximally folded in its best conformation, it's just better. And so what's happening here in this interior is that you are helping those hydrophobic residues move from the exterior to the interior. And so anyway, so it goes through this cycle. And again, the protein doesn't transition from one to the other. It's just that one side is active and helps fold, then the other side will get a protein and is active and helps fold. And so this ring here will bounce between the top and the bottom there. So let's look at some numbers. So the GROW-LES complex gives proteins an opportunity to fold correctly, but it doesn't really actively rearrange them. It just helps remove them from the solvent and allow those hydrophobic groups to help find their way to the interior. So about 5% of a substrate molecule fold per reaction cycle. So that's about 14 cycles would need to fold half a protein sample, and that costs 98 ATP. That seems like a lot of ATP to try to get a protein folded. But if you look at the cost in synthesizing the protein, this is a drop in the bucket. So once you've expended all that energy making a protein, a few hundred ATP to get that protein folded is a small price to pay for all that energy that you've already exerted. And so most of the substrates that these work on are between 20 and 60 kilodaltons, and they tend to have these extended beta sheets that are difficult to fold. So we talked a little bit earlier that beta sheets tend to be slower in folding, where alpha helices tend to fold a lot more rapidly. Now, this GROW-LS complex is actually prokaryotic, so we do have a eukaryotic one called Tri-C. It does have double rings of 8 subunits. Those 8 subunits are different, where in the GROW-LS, they're actually, there's 7 subunits, and those are all identical. So in the eukaryotic, there's 8 subunits, and they're different, but it functions similarly. And it helps fold about 10% of eukaryotic proteins. So they've done studies and figured out how much of our eukaryotic proteins interact with these chaperones, and about 10% of them need a little help to get folded correctly. So this was the last question that I asked, well, what happens if the protein never gets folded correctly? Well, the protein could be completely denatured and not really have any structure, or it could adopt a misfold. So what we have up here is an example of a native protein that for some reason either misfolds or undergoes a conformational change and never gets back to its native form, or is never completely denatured. And then this one here can undergo some further conformational changes. And so you can see what it's doing is it's exposing these beta sheets there. And these beta sheets can then associate with other beta sheets, and then those beta sheets can associate with other beta sheets. And what you can do is start to form fibers there. And these fibers are referred to as amyloids. And so misfolded proteins are often referred to as amyloid plaques. And so native beta amyloid is a cellular globular protein. And I can't remember if this one, I don't think this one has a known natural purpose, but it can undergo some misfolding and then aggregate, and then you get these fibers. And these fibers can be problematic. Let's just look at some diseases and then we'll look at what they do. So Alzheimer's disease is made by the defective protein amyloid beta protein. ALS, or Lou Gehrig's disease, is you get the defective protein superoxide dismutase that actually aggregates into these fibers, and you can see the other ones down there. And so these proteins misfold, and then as they misfold, they tend to aggregate together. And when they aggregate, they make a very hydrophobic and very stable structure that is hard for the body to get into and break down. And so you get these plaques. And so here's a plaque in here. And as these plaques start to form, they start to cause neuron death around there. And so this amyloid beta protein, it actually comes from a 770 amino acid membrane-bound precursor, and they're not sure the function of it. And then we actually have enzymes, secretus, let's try that again, secretases that cleave a small fraction of this membrane-bound protein and get the amyloid B right there. So they're not sure what this does. I'm not sure what the full protein does, but these enzymes will cleave off a fraction and give you this amyloid B, and then that amyloid B can misfold and start to form these plaques. And so it's actually thought that the neurotoxic effects probably start in the early states of the disease. You just really don't see it until they really get advanced. And there are people with certain mutations that develop early onset. So people with Down syndrome have an extra copy. I believe it's trisomal 21 that they have. And so there's an extra copy of this beta precursor gene in each cell. And so they have extra beta precursor. And so they tend to get Alzheimer's at an early age. And so it's thought, well, if we could inhibit these secretases, then they wouldn't be cleaving off this amyloid beta. And so there's been lots of trials in trying to make inhibitors for that, and lots of really promising results, but nothing that's made it to the market yet. They tend to fail in the phase 3 trials for some reason. And so protein folding, some proteins are just inherently unstructured and can exist in multiple conformations. So you can refer to them as IUPs, intrinsically unstructured proteins. So they don't have a defined structure. It actually depends on the physiological condition or the other proteins that they're involved in, or sorry, interacting with. So you can refer to them as kind of metamorphic. They can change it. And so here is an example of one. And you can see that it's got some beta sheet and an alpha helice, but then it can go to all beta sheet. And depending on the structure, it has different functions. I just wanted to share that with you because it seems like these misfolded proteins kind of have that same type of possibility that they can actually adopt different conformations. We're just not sure what their purposes are. And now there's also known as infectious prion diseases, and these are somewhat related to these protein folding diseases in that you get misfolded proteins that aggregate and eventually cause problems and death. And so there's disease known as scrapie, bovine spongiform encephalopathy, BSE, also known as mad cow disease, Kuru, which is an interesting disease. If you want to read something fun, go look up Kuru on Wikipedia. It has to do with human cannibalism. And then you got Creutzfeldt-Jakob disease in humans, which is thought to be very similar to Kuru disease and also BSE, mad cow disease. What's interesting about these is the infectious agent that can be transmitted from one species to another is proteins that aren't coupled with nucleic acids, not virus related. So meaning you're not transferring genetic information from one organism to another like we're experiencing with COVID-19 now. You're just transmitting a protein to another organism, and that protein by itself will cause other proteins to misfold. So these are known as prions, or protein infectious agents, and they're mostly hydrophobic proteins produced from a normal cellular gene. And we're not sure what that gene functions as, meaning that if you knock this gene out in mice, the mice appear normal. So for some reason, we got this protein floating around, and when it's in its correct conformation fold, it does nothing. When it misfolds, it's going to aggregate and cause neurological problems and eventually death. Okay, so one of the things that they found was that if you feed infected protein into other animals, they'll get infected. So one of the ways that they found this out was they feed the byproducts of cattle slaughter, meaning bones and brains and nerve tissue, to other cows. Nothing goes to waste on a farm. So when they pluck the feathers off of a chicken, they process those feathers and feed it back to cows. They call that feather mill. When they butcher the cows and get the bones, they process those bones and feed it back to chickens as bone meal to help their eggs have more calcium. So they used to cycle a lot of this animal waste and just put it back into feed, because essentially it's just protein, and animals that are growing on a farm need protein. But they included some sick cows, and these sick cows had misfolded proteins, and they included the brain stem and brains of these cows. And when they included that in the feed, they noticed that they started getting a lot more sick cows. And so lots of studies went on, but eventually this is the normal folded protein, and this is the misfolded protein, again, that has this beta sheet. But if you introduce this protein into a cow, it will start to develop these mad cow disease. It doesn't need DNA, it just needs proteins. And then what it's thought is that this protein causes other proteins to misfold, and kind of catalytically, you start to get misfolding of lots of proteins. Again, this happens in the brain, and that's not a good place to start to get these plaques or these fibrils. So you get these long aggregations of these fibers. They're virtually indestructible, long, twisted, stacked beta sheets. And again, your body can't process them. They get in your nerve cells and eventually get so long and bulky that they interrupt those cells and they tend to die. And you get neuronal death and the diseases that we saw, Alzheimer's and ALS and Parkinson's disease, mad cow's disease, Creutzfeldt-Jakob disease. Anyway, so make sure that your proteins are folded correctly. And we'll leave it at that. That is the end of chapter 6. Thanks.